Hi, thank you for visiting. I'm Satoru, and I'm a postdoc in Massimo Scanziani's lab at UCSF. In this poster, we address how motion induced on the retina by the animal's own saccade can be distinguished in visual cortex from motion occurring in the external world. Saccades are rapid eye movements performed by many animals throughout the animal kingdom, including us humans. And while these movements are very useful, they also pose a big challenge. And the challenge is to maintain a stable representation of the world despite these saccades constantly shifting the visual scene across the retina, creating motion. How do animals differentiate motion that occurs in the external world from one that is induced by the animal's own saccade? We addressed this question in mouse primary visual cortex, or V1. To find out whether the response to saccade-induced motion is different from the response to external motion, we made extracellular recordings in V1 of awake, head-fixed animals under two conditions and compared them. In the first condition, animals made spontaneous saccades on a stationary vertical grating. Because saccades are mostly horizontally head-fixed animals, these vertical gratings induce a strong motion on the retina during saccades. In the second condition, we shifted the same vertical grating in a manner that mimics visual changes caused by real saccades. In other words, this external motion was approximately matched in direction, amplitude, and speed to the real saccades in the left, and we call these pseudosaccades. And so on one hand, we have motion on the retina induced by saccades, and on the other hand, we have the exact same motion on the retina, but one that is induced externally. Both of these conditions induce directionally selective responses in V1 neurons. And these are example neurons that show such direction selectivity under each of these conditions. Now we know that moving gratings in general induce directionally selective response in V1 neurons. And so this response to saccades may simply reflect the response to motion of the grating on the retina induced by saccades. If this were the case, the neuron should behave in the same manner regardless of whether the motion results from saccades or pseudosaccades. In other words, the direction selectivity of neurons under these two conditions should correlate. Interestingly, in contrast to this expectation, there was a lack of correlation, and many neurons reversed their preferred direction during saccades. Thus, direction selectivity is scrambled during saccades, and saccades and externally occurring motions induce distinct patterns of activity in V1. Accordingly, because of this scrambling, if we train a linear classifier on the response to pseudosaccades to discriminate the different directions of motion, the decoder fails to generalize to real saccades and performs very poorly. So what's causing the scrambling of direction selectivity? We hypothesize that this is due to a non-visual input that enters V1 during saccades. Indeed, when we deprive the animals of visual input, in this case by pharmacologically silencing the retina, we found that many V1 neurons still responded to saccades and discriminated the direction. In the middle is an example neuron that strongly prefers temporal saccades over nasal saccades, and on the right is the average peri-event time histogram for preferred versus non-preferred direction. And furthermore, the timing of the response, which precedes the actual saccade onset by a couple of hundred milliseconds, suggests that this non-visual input is at least in part a copy of the oculomotor command, or an afference copy. And we show that this non-visual input comes from the pulvinar, a higher order nucleus of the thalamus that sends extensive projections to V1. By silencing the pulvinar in animals deprived of vision, we were able to eliminate much of the non-visual saccade-evoked activity in V1. So is this non-visual input from the pulvinar the cause of the scrambling of direction selectivity? Well, if this were the case, then by silencing the pulvinar during saccades in otherwise unmanipulated animals, that is non-blinded animals, we should be able to eliminate the scrambling since all that's left in V1 during saccades should be the visual input. In other words, now the direction selectivity during saccades and pseudosaccades should correlate. And indeed, that's what we see. Furthermore, consistent with this result, the linear classifier trained on the response to pseudosaccades was now able to decode the direction of motion on the retina, regardless of whether it was caused by saccades or pseudosaccades. Thus, our study shows that motion induced by saccades and the motion occurring in the external world induce distinct patterns of response in V1, and this differential response is due to the scrambling by non-visual input from the pulvinar. Furthermore, we propose that this scrambling may prevent the downstream from extracting information about motion on the retina induced by saccades. That's a quick summary of the story, and I hope you'll enjoy my poster. If you have further questions, please feel free to contact me, and I'm looking forward to your feedback. Thank you.